So this week we're going to be talking about DNA and how DNA is an important piece of evidence used in order to tie criminals to a crime scene. So in this case, when we're talking about DNA, we want to make sure that we understand the foundations and the basic information associated with DNA. So if you remember from biology class, there was an emphasis on making sure that we understood the components of DNA and how that kind of fit into the world of biology. So this is just a brief introduction to what you probably already know, but we want to make sure that we touch up on some information that's important for this particular class. So in terms of DNA, it stands for deoxyribonucleic acid, and it is made up of nucleotides. And nucleotide is made up of a simple sugar, a phosphate group, and a nitrogen base. There are 4 nitrogen bases associated with DNA: adenine, guanine, cytosine, and thymine. In this case, when we're talking about adenine and guanine, these are going to be our purines. They're double ringed. Easy way to remember that is that there are 2 N's in each one of these words, whereas there's only 1 N in cytosine and thymine. Your pyrimidines are going to be your cytosine and thymine. They're single ringed, and again, an easy way to remember that is the number of N's. So if there's 1 N here and there's 1 N here, so that makes them single ringed. Now, in order to determine whether or not you have purines or pyrimidines, an easy way to remember that is the Y in pyrimidines. So cytosine has a Y in it, thymine has a Y in it, but there are no Y's in adenine or guanine, so your pyrimidines are going to be your cytosine and thymine. So hopefully, again, this is kind of a review of things that you've already learned in biology. So these are your double and single ring. This is available to you on Schoology, so you're more than welcome to take a look at that if you'd like. Now, in terms of DNA, we also know that DNA can be calculated based off of the number of nitrogen bases and also what they pair with. So Erwin Chargaff came up with an idea that the amount of A is going to always be equal to the amount of T, and the amount of C is always going to be equal to the amount of G. So knowing that, we should be able to calculate any number of percentages associated with the nitrogen bases so long as we know one of them. So let's say, for instance, that thymine is equal to 10%. Well, if thymine is equal to 10% and adenine and thymine are equal to each other, that means that adenine and thymine both must be 10%. So that adds up to 20%. Now, how do we figure out cytosine and guanine? Well, if cytosine and guanine must account for the other 80%, because you have 100% of the DNA that must be accounted for, cytosine and guanine must account for the other 80%. Since there are 2 of them and they must be equal to each other, an easy way to figure that out is just simply divide by 2, so that each one of them is going to be worth 40%. Okay, so if you're not 100% sure about that, there are going to be some practice problems associated with making sure that we understand the different amounts. So this is an example of a DNA strand. So DNA is double-stranded. So we can see that there's 1 strand here, and then there's another strand here. And we also know that there's complementary base pairing occurring with our nitrogen bases. So that A is always going to be paired with T, G was always going to be paired with C, G is going to pair with C in this case, and then A is going to be paired with T in this case. And so this is a very, very easy way to kind of see what DNA is made up of. It's made up of these 3 components of the deoxyribose, the phosphate group, and the nitrogen base, which all 3 of those will make up this nucleotide here. Hydrogen bonds exist in between each one of these, and that these hydrogen bonds are really important in making sure that they keep that DNA strand together. So that if we want to replicate our DNA, if we want to copy DNA, if we want to make sure that we're able to analyze DNA, we must break those hydrogen bonds, and we'll talk about that later. So you have two scientists that are going to be really important in terms of the discovery of DNA, Watson and Crick, and they're the ones that made up the model of DNA. And so they're the ones that came up with this complementary base pairing of A's, T's, C's, and G's. And so A will always pair with T, C will always pair with G. And this was really important in making sure that we're able to come up with a way to synthesize DNA and come up with a way to be able to copy DNA, because this kind of laid the foundation for all that information. So that's what we see here in terms of adenine pairing with thymine and cytosine pairing with guanine.